Olá, boa tarde a todos. 2h30, então já vamos começar aqui com o nosso webinar. Enquanto vocês vão se conectando, o pessoal vai se conectando, vou fazer uma breve introdução aqui sobre o nosso webinar, que hoje vai falar sobre clonalidade, tá bom? Sejam muito bem-vindos ao nosso webinar sobre clonalidade. Esse é mais um encontro de uma série especial que estamos promovendo junto com a Bio-Techne. Para quem ainda não me conhece, eu sou a Larissa, faço parte do time de marketing aqui da Enterprise. Vou fazer uma introduçãozinha básica aqui sobre o nosso webinar, os temas importantes. Em seguida, eu vou passar a palavra para os nossos convidados, né, que vai ser as pessoas responsáveis por conduzir o conteúdo hoje. Então, hoje iremos falar sobre clonalidade. Então, como clonality is king, o clonalidade é a chave. Abordaremos alguns tópicos importantes, então, dentre eles, a importância da geração de linhagens celulares clonais de alta qualidade para pesquisa e produção biológica. Também falaremos um pouquinho sobre a visão geral das funcionalidades do Pala, além de estratégias para otimizar o fluxo de trabalho no desenvolvimento de linhagens celulares no seu laboratório, tá bom? Então, como vocês já sabem, ou se é o primeiro webinar que está participando conosco, só para contextualizar, a Enterprise é a representante e distribuidora exclusiva da Bio-Techne aqui no Brasil, exceto as regiões Nordeste e no Paraná. Nós possuímos uma equipe completa que está preparada para dar todo o suporte para vocês necessário, então desde a parte de logística, importação, documentação, além de dúvidas técnicas sobre os equipamentos e kits, tá bom? Gostaria de trazer um lembrete importante também, que hoje esse webinar ele vai ser conduzido em inglês, então caso vocês precisem, o Zoom oferece uma ferramenta, né, a opção de legenda automática, então é só aqui no rodapé, vocês clicarem ali na legenda ou onde está a palavra CC, na barra inferior, que vocês conseguem ativar de acordo com a preferência, tá bom? Agora eu gostaria de apresentar para vocês a Laure, que ela é a nossa líder técnica da Bio-Techne na América Latina, para se apresentar um pouquinho e logo depois seguimos com o conteúdo do nosso webinar. Seja bem-vinda, Laure.
Obrigada, Larissa. Boa tarde a todos. Obrigada a vocês aí que estão interessados nesse webinar. Então, eu sou a Laure, meu trabalho aqui é como líder técnica regional para toda a América Latina. Então, eu sou responsável pelo suporte técnico-científico do portfólio da Bio-Techne em toda a América Latina. Eu trabalho muito em contato com a Enterprise, que é nosso distribuidor, né, exclusivo aqui no Brasil, com exceção das regiões da região Nordeste e do Paraná. Eu tenho bastante experiência na pesquisa acadêmica, principalmente nas áreas de biologia celular e molecular, e há 6 anos, né, eu venho trabalhando aí como especialista de produtos e aplicações, e há 2 anos e meio, agora aqui, responsável pelo suporte técnico-científico da Bio-Techne na região, tá? E hoje eu tenho muito prazer, estou muito feliz aqui de fazer esse webinário para vocês que vêm acompanhando essa série de webinários com a Enterprise. Vocês veem que na maioria das vezes sou eu que apresento, e aí para o pessoal não se enjoar, eu convidei, né, um colega, que é o Ryan McComb. Ele é gerente de produtos da Bio-Techne e gerente de produtos da linha do Pala, do nosso dispensador de single cell, que nós vamos falar hoje sobre essa tecnologia, né? O Ryan, ele é formado, tem bacharelado em biologia celular pela Universidade da Califórnia em San Diego, mestrado e doutorado pelo Instituto Keck em Claremont, também na Califórnia. Ele fez pós-doutorado na Universidade de Stanford no departamento de genética, e antes de se juntar à Bio-Techne como gerente de produtos para os dispensadores de células únicas, né, ele trabalhou como cientista e também gerente de produtos numa empresa de biotecnologia chamada Recursion Pharmaceuticals, tem uma vasta experiência, tanto na academia quanto na indústria, com desenvolvimento de linhagens, com desenvolvimento de drogas e small molecules para uso farmacêutico, tanto em doenças cardíacas quanto em doenças neurodegenerativas, né? E tem sido um grande recurso para a gente na empresa, para os clientes também, para essa linha, né? Para essa parte de otimização e validação de geração de linhagens celulares utilizando o Pala. Tá bom? Então, espero que vocês aproveitem, como a Larissa falou, apenas reforçando, esse webinário vai ser em inglês, né? Mas aqui o Zoom tem uma ferramenta que vocês podem encontrar se vocês precisarem de apoio, que é a tradução instantânea. E ao final a gente vai ter uma sessão de perguntas aí com o Ryan, tá bom? Aproveitem.
Thank you for joining our Clonality is King webinar. My name is Ryan McComb, and I'm the Product Manager for Bio-Techne's Single Cell Dispensing Instruments. Prior to Bio-Techne, I've had the opportunity to develop clonal cell lines for basic research and product development, and I will be sharing with you my experience with the Pala platform and how I believe this platform can be a game changer for your research. First, I will talk about some challenges and bottlenecks in cell line development. Then I will introduce you to Bio-Techne's Pala system, and finally I will run through some key applications and workflows from some of our customers that have integrated this technology into their daily use. With the advancement of new technologies, cell line development has entered a new era with a high demand for easier methods and tools for generating stable clonal cell lines. Accessibility and adoption of gene editing tools like CRISPR and the expansion of iPSC technology have led to more labs generating cell lines for academic research and drug development. Also, the growing demand for new drugs continues to create a need for more productive and stable cell lines for manufacturing biologics and cell therapies. In most cases, clonal cell lines must be generated from a single cell isolated from a transfected pool and then screened for the desired mutation and phenotypic characteristics. The importance of generating clonal cell lines cannot be emphasized enough. This is because clonal cell lines enable greater control to generate reproducible data. In manufacturing, clonal cell lines provide more predictable growth rates, production titers, and stability for manufacturing biologics. Rigorous characterization is necessary for any type of manufactured biologic. The FDA, EMA, and International Council for Harmonization all have guidance on cell line characterization and quality control and require proof of cell line clonality for manufacturing recombinant proteins. Generating single cell-derived clonal cell lines is not an easy task. Many challenges and bottlenecks still impede research productivity. These include low gene editing and transfection efficiencies, low single-cell survival due to difficult-to-express phenotypes or transgenic proteins, all resulting in excessive screening burdens and labor requirements. Traditional approaches for single-cell isolation have included FACS and limiting dilution using manual or automated pipetting. These techniques suffer from a variety of drawbacks. For FACS instruments, sorter-induced cell stress caused by the high pressures used in traditional FACS instruments can reduce cell viability and outgrowth. Instruments can be difficult to operate and require specialized training, and experiments can be very time-consuming for setup and execution, while also requiring large amounts of starting material. For limiting dilution, poor single-cell deposition efficiency results in low success rates and a high screening burden for identifying wells with single clones that also have the mutation of interest. Limiting dilution also has no way of enriching target cells with fluorescent labels or viability stains. These drawbacks result in lost time and productivity. I will now introduce you to Bio-Techne's Pala, our solution for automated fluorescence-based single-cell dispensing. Pala has been specifically designed with the bottlenecks and inefficiencies of the traditional approaches I just mentioned in mind. The Pala has compact design for use in standard tissue culture hoods or on a lab bench and provides a fast instrument startup, cell dispensing, and shutdown. The software is easy to use and provides flexibility for a wide variety of cell types and fluorescent dyes. The Pala has a low cost of ownership when the instrument, consumables, maintenance, and time are factored into consideration. Pala has numerous advantages over traditional facs. For starters, the Pala uses less than 2 psi of pressure compared to traditional fax instruments that may use up to 70 psi of pressure for cell sorting. To put this into context, a typical car tire is pressurized at around 35 psi. The setup time for Pala is a fraction of the time as fax. The simple software allows for users to be trained within a single 1-hour training, and once trained, users can execute a sort within 7 minutes. The Pala also allows for very low minimal cell inputs, and when a sample is analyzed and sorted, the single cell dispensing volume is 1 microliter compared to an aerosol-sized droplet that fax uses. Pala is also very easy to clean and sterilize for cell line development needs. Bio-Techne's dispensing technology is enabled by 3 core technologies: microfluidics, flow cytometry, and liquid dispensing. When a sample is loaded into a microfluidic cartridge, the cartridge is then loaded into the instrument. The instrument pressurizes the cartridge with 1.8 psi of pressure, and cells are directed down the microfluidic channel where they are interrogated with up to 2 excitation lasers. Light scatter and fluorescence emission signals are used to analyze and sort cells of interest. Once the target cell of interest is detected, a high-speed valve opens and pushes the cell out of the cartridge through a nozzle and into the microtiter plate, leaving the non-gated cells behind. The microfluidic cartridges that the Pala uses are made of COC plastic, a highly advanced optically clear polymer plastic. Each cartridge is individually packaged in a sterile wrapper and can be loaded with up to 600 microliters of cell sample and reloaded with sample over the course of a single day. Due to the closed design of the cartridge, your cell sample does not come into contact with the instrument, making sample-to-sample carryover impossible. The Pala comes in 2 optical configurations, either a dual 405 and 488 nanometer laser configuration or a dual 488 and 561 nanometer laser configuration. This offers a variety of fluorescent channel options that Pala can detect. For example, the 405-488 model can detect up to 11 fluorescent channels with 2 light scatter channels, while the 488-561 model can detect up to 9 fluorescent channels with 2 light scatter channels. Other notable features include a quick dispensing time for 96-well plate of less than 2 minutes, or 6 minutes for a single 384-well plate. Most common dyes and fluorophores are accommodated by the Pala. For genomics applications, many of our customers use the 405-488 option, since this allows for common nuclear stains such as DAPI and other violet dyes. Customers who prefer using fluorescence proteins or dyes in the far red spectrum benefit from our 488-561 option, since this allows labels such as M-Cherry and PE-based tandem dyes. The Pala is also compact and portable and weighs just 35 pounds, making it possible to move in and out of your biosafety cabinet and keeping your bench space flexible. Now let's talk about some of the performance capabilities of the Pala. The Pala can dispense cells in both 96-well and 384-well plate formats. In recent validation experiments, we used both beads and cells to test the instrument's performance. In this experiment, we dispensed 72 plates split between 6 instruments. Pala's average 96-well plate deposition accuracy was 92.5% of wells containing a single bead, while only 3.7% of wells contained 2 or more beads. While Pala's 384-well dispensing average was 85.3% of wells containing a single bead, and again only 3.7% of wells containing 2 or more beads. When we look at cell dispensing accuracy for CHO cells grown in suspension and A549 cells grown as adherent, we demonstrate that the Pala shows an 80% deposition accuracy of wells with single cells detected and less than 3% of wells containing multiple cells per well. Compared to limiting dilution, we show that on average, the Pala obtains 3 to 4-fold more single cells deposited per plate. However, with limiting dilution, this varies depending on the accuracy of the cell count, while Pala can tolerate a larger range of cell concentrations without reducing single cell deposition accuracy. How do we confirm a single cell is deposited in each well? We found that for reliable cell deposition measurements, it is useful to obtain images from both fluorescence and bright field channels. We recommend using viability dyes such as Cell Tracker. This not only gives you the ability to gate out dead cells when dispensing with Pala, but also provides fluorescence confirmation during plate imaging. Bright field is necessary to provide data on cell shape and translucence, and combined with fluorescence data can help us to identify single cells from doublets, as well as differentiating between plate debris. When we look at outgrowth rates of CHO cells and A549 cells between Pala and limiting dilution, we found that they are almost identical. This indicates that Pala's handling of CHO and A549 cells is not inducing any more cell stress than manual hand pipetting. To demonstrate the time savings of using Pala over limiting dilution, I will now walk through the following example. With target cells varying in proportion of the total population at 1%, 10%, and 100%, we show that the Pala would require half of a 96 well plate to obtain 30 clones, assuming an 80% deposition and 80% single cell outgrowth rate. If 100% of the cells in the population are being dispensed, the dispensing time would take about 1 minute. If 10% of the cells in the population are being dispensed, the dispensing time would take about 6 minutes. And if 1% of the cells in the population are being dispensed with the Pala, the dispensing time would take about 35 minutes. Since only half of a 96 well plate is required for each of these populations, regardless of the proportion of cells dispensed, the plate screening time is minimal and only 5 minutes if assuming your imager can screen a 96 well plate in about 10 minutes. If we now look at limiting dilution, which does not allow for fluorescence-based selection, we find that this experiment quickly becomes unfeasible at lower proportions of target cells. For example, the 10% target cell abundance would require 13 96 well plates to obtain 30 clones, while the 1% target cell abundance would require 130 96 well plates to obtain 30 clones, assuming a 30% single cell deposition rate and an 80% outgrowth rate. Handling this many plates is only half of the problem, since screening those plates using an imager will take another feat of endurance, since it is impossible to know beforehand which wells received a single cell. Now I'm going to talk through some of the applications and workflows from our customers. As I mentioned previously, iPSC technology paired with gene editing creates a powerful tool for studying disease and developing new drugs. Cells taken from diseased patients can be differentiated into iPSCs. These cells can be gene edited to introduce desired genetic corrections or changes that allow for the ability to study genetic and phenotypic relationships in an unprecedented way. Additionally, this technology can be used to generate differentiated, corrected cells that can be reintroduced into the patient as a cell therapy. For example, using our technology for stem cell development from our customer at the NIH at the Stem Cell Translation Lab, these authors aim to develop tools to ensure safe and robust long-term growth and functional differentiation of human pluripotent stem cells. They used high-throughput screening to identify small molecules to improve the viability and outgrowth of the cells. They identified a combination of chromin-1, imricasan, polyamines, and trans-integrated stress response inhibitor, and dubbed this the CEPT cocktail. This cocktail primarily acts by blocking cellular stress pathways, and the authors show that the CEPT cocktail provides viability improvements for several applications, including cell packaging, cryopreservation, embryoid body and organoid formation, single cell cloning, and genome editing. They successfully performed this research using Bio-Techne's single cell dispensing technology. In a second paper, a collaboration between Cytovia Therapeutics, Selectus, and UCSF demonstrated a workflow using TALEN technology to generate dual-edited IL-15 knock-in and TGF-beta receptor 2 knockout iPSCs. These cells were then able to produce enhanced iPSC-derived natural killer cells for cancer immunotherapy. This work importantly shows that these iPSCs could be efficiently differentiated into natural killer cells that showed enhanced autonomous growth and are resistant to suppressive TGF-beta signaling in the tumor microenvironment. These two studies that I just highlighted show the power of utilizing Bio-Techne's single-cell dispensing technology in human stem cell development. Shifting applications, CHO line development is the bread and butter of biologics manufacturing for clinical applications. Hundreds of clones must be screened for each manufactured product to identify those with the highest production yield, stability of product, and stable growth rates. To develop a robust manufacturing process, CHO cells are first transfected with an expression vector containing a gene of interest. Cells that are successfully transfected can be selected for using antibiotics or other selection methods. Then cells must be single-cell cloned in order to develop a clonal cell line that meets the required characteristics for product specificity, yield, stability, and scale-up requirements. In a recent paper by our customers at AstraZeneca, it was demonstrated that the Pala was able to generate high rates of single-cell clones as determined with plate-based imaging. The authors reported a 96.3% single-cell deposition efficiency in 384 well plates and a 3.7% multiple cell per well rate. This mirrors the data I presented previously for 384 well dispensing rates. Additionally, you can see in the top figure A, B, and C for the easy-to-express protein that Pala and FACS show comparable performance of clones for productivity and growth rates. However, with a difficult-to-express protein, figures A, B, and C below show that Pala produced clones that outperformed FACS in productivity while maintaining performance and growth rates. This is evidence that Pala's gentle cell handling provides an advantage over FACS for single-cell cloning of sensitive cell types. In our final application, Bio-Techne's cell dispensing technology has been successfully adopted for many antibody development groups deploying traditional hybridoma technology and newer single B-cell isolation and NGS sequencing approaches. Labs performing antibody discovery and development are often handling multiple samples per day. Some of our customers handle up to 10 different samples in a day. Pala provides a simple solution for dispensing single hybridoma cells in 96 and 384 well plates, and the cartridge-based dispensing does not allow sample carryover. Additionally, Pala's portability is ideal for co-location in the lab that the immune cells are being harvested. In summary, we've shown that Pala offers significant time savings for generating clonal cell lines with fluorescence selection over FACS and limiting dilution. Pala dispenses single cells with high accuracy and generates clones with high outgrowth rates. We showed that up to 4-fold more single cells can be deposited per plate than limiting dilution, and that the proportion of cells that grow into colonies were comparable to limiting dilution. Finally, we showed that Bio-Techne's cell dispensing technology has been adopted and proven in many cell line development and engineering workflows, including in workflows for iPSC gene editing, CHO cell biologics manufacturing, and antibody discovery and development. Thank you for listening, and please reach out if you have any questions or would like to see the Pala in action at your lab.
Obrigada, eu gostaria de Agradecer aí ao Ryan, que está disponível e trazer esse conteúdo para a gente, também a Laura. Tá? Agora a gente vai começar com a sessão de perguntas. São as perguntinhas que vocês enviaram para a gente durante a inscrição. Nós selecionamos algumas mais comuns. Se você não enviou e gostaria de fazer alguma pergunta, peço que você deixe aqui no chat. A gente vai tentar responder o maior número possível de perguntas. Se a gente não conseguir, a gente entra em contato com vocês por e-mail, tá bom? Então, começando aqui com a primeira pergunta que a gente recebeu, foi a seguinte. Eu vou fazer a perguntinha aqui em português e a Laura vai passar para o Ryan, tá bom? Então, a primeira pergunta foi: quais são os principais desafios ou limitações na otimização da dispensação de single cell com o Pala?
Obrigada, Larissa. Então, pessoal, eu vou traduzir aqui, tá? Pro Ryan. First of all, I would like to thank you very much for your time, for being here with our Brazilian customers. I'm very happy to have you here, Ryan. So I will start with our first question that was sent by a customer. What are the main challenges and limitations on the optimization of the single cell dispensing with Pala?
Thanks for the question. Can you hear me okay?
Yes, we can.
Okay, good. So with single cell dispensing, the instrument can very accurately dispense single cells in the well. However, sometimes different cell types need certain conditions to help them grow into colonies. So often for certain cell types, you may need to have a conditioned media or certain growth factors to help grow those cells into colonies. So dispensing the single cell into a well is part of it, but the other part is getting that to grow into a colony, which that requires some optimization. And we recommend when doing that to test your cells out with different conditions, both with the Pala, both with limiting dilution to help you understand the variables that are affecting the outgrowth of the cells. And sometimes if you're doing gene editing, for example, it can be some of the edits that you make can affect the viability of single cells and their ability to grow. And so that's another consideration you have to make.
Okay, great. Thank you. So it's not only the Pala, the single cell dispensing, but all the workflow that comes after that, right? That makes it good for the cell. So Larissa, we have another question. Is that right? Can I translate?
A próxima perguntinha aqui que o pessoal mandou foi a seguinte: qual o mínimo de porcentagem de uma população alvo de células que o Pala consegue dispensar?
Tá. So the next question, I think it's a pretty common question here. How low percentage of a target cell population can Pala dispense? I think when we speak about rare cells, how low it's efficient to use with the Pala?
Yeah, good question. So we've tested here in San Jose, where I'm based. We've tested concentrations down to 0.1%, and are effectively able to sort those out and confirm that we've sorted those cells out of the cell mix. We do have customers that have reported going much lower than that, but I don't have that validation data to share. But we have definitely validated 0.1% populations, and Typically, our customers are not going that low, but the Pala is capable of, as long as your cell is, you can find it with fluorescence. The Pala should be able to sort it. It just will take longer depending on the rarity of the cell.
Right. Ok. Thank you. Thank you.
Próxima perguntinha foi a seguinte: quais são os limites de tamanho de células e partículas que o Pala consegue dispensar? E qual a maior e a menor célula e partícula?
Okay, next question. It's also a common question when we talk about Pala. It's about the size. So what are the limits of the size limits that Pala can dispense when we talk about cells or particles? What are the biggest and the smallest cell or particle that Pala already have dispensed?
Yeah. The cartridges that we make are specifically designed for mammalian cells because the size of the channel in the cartridge is about 50 to 150 microns. So you really can't go higher, I would say, than 35 micron cells because they'll start to kind of clump up in the channel. In terms of the smallest, that's more of the ability for the instrument to optically detect that small cell, which we test the instrument down to 3 micron particles, and the instrument can easily detect 3 micron particles. So I say between about 3 and 35 microns, which covers everything from nuclei all the way up to large, IPS cells, for example, are about 20 microns. They're fairly big. So you have quite a range. CHO cells are about 13 microns on average. T cells are like 6 microns. So we can cover quite a range of cell types. What we can't do very well is bacteria or like organoids, multiple cells clumped together.
Okay. Ok, cool. Thank you.
Próxima pergunta foi a seguinte: a dissociação de tecido cerebral frequentemente resulta em células frágeis e com baixa viabilidade. Como o Pala lida com essas amostras delicadas para garantir a viabilidade?
Okay, then this is a very good question. So dissociating brain tissue usually results in fragile cells with low viability. How Pala can manage that? How Pala can manage those delicate samples to ensure that we are gonna have viability after sorting?
Yeah, good question. So we actually have an application note that Lori can share after the call, where we actually did experiments with brain single nuclei that were sorted for single cell 10x genomics transcriptomic analysis. So we can share that later, but in terms of the gentleness, so Pala is less than 2 psi of sorting pressure, and it gives the ability for, because it's a low pressure, you have less cells that will die during sorting, whereas traditional FACS instruments, we often hear from our customers that they tried sorting like brain tissue or tissue from their animals using the traditional sorter, and nothing survived. So I can't guarantee that the Pala is going to work for every sample. We haven't thoroughly tested it across all different tissue samples, but the fact that we're sorting at low pressure at low speed gives you a better chance of being able to sort those viable sort viable cells from primary tissue. And we do have customers that do use that workflow for preparing their sample before, say, 10x genomics sequencing, so that they can get a better representation of healthy cells.
Yeah, and please correct me if I'm wrong, but from the experience I've been having here, it's not just with this specific type of cell, like from brain tissue, but all of type of cells that we have faced this issue with low viability after FACS. We have been most of the time successfully able to sort and have a good viability after our dispensing with Pala, right? So not only for this specific brain tissue, but others type too.
Yep. Yes.
Cool. Thank you.
Mais uma perguntinha aqui. Quais são as vantagens do uso do sistema automatizado de células únicas em comparação aos FACS nos sorteios de neurônios? Isso, sorteios de neurônios ou células gliais para perfis transcriptômicos. Outra pergunta boa.
Okay. So I think this question is related to the last one. What are the advantages of using the Pala automated dispensing system in comparison with FACS for the sorting of neurons and, I don't know how to say exactly this, glial cells to study transcriptomic profile?
Yeah, so one of the things that happens during FACS is that because cells are being sorted at high pressure in FACS, when they come out of the nozzle, there's a decompression that the cells undergo. So there's that kind of shock that they go through when they're being sorted with high pressure. The other consideration is the nozzle size. That, if you're using a FACS instrument, you're generating very, very small droplets with very small nozzle, which causes stress on the cells as well. And so if you look up the 10x genomics sample workup procedure, they recommend to use the largest nozzle size and the lowest PSI of pressure that you can when you're using FACS sorting for preparing samples for 10x, for example. So the Pala, because you're running it at very low PSI, less than 2 PSI, and the nozzle size generates a 1 microliter droplet, we fulfill those requirements that are recommended in the 10x workflow. When you're looking at the workflow they recommend. So the large nozzle size and the low sorting pressure are two recommendations, and the Pala has that capability. If you're looking to enrich a sample, the Pala also has a feature called bulk sorting, so basically the idea there is that we can sort faster than one cell per second. We can sort at a rate that's faster to help enrich your sample. So if you're trying to enrich a population from, say, like a 1% or a 5%, and you want to get more of a population, like, say, in the 40 to 60% range for your 10x workflow, so that you're not wasting a lot of reads on cells that you're not interested in. The bulk sorting capability, which I didn't talk about in this presentation, but it does have the capability of sorting faster and enriching a sample. So if you need, say, like 5 to 20,000 cells to go into your 10x workflow, there's that capability as well.
Yeah, nice. Thank you. And it was very good that you mentioned the bulk sorting mode. because I took a note during the presentation here. If you could quickly just go through the 3 modes that Pala does. Not only we focus today on the single cell sorting, but we have 2 other modes that we can use the Pala too. So it also makes the instrument more flexible in the applications, right? If you can comment on that too, it would be very nice.
Yep. So single cell dispensing that's the primary mode. That's the one that the instrument was primarily developed for dispensing one cell into a well for cloning. Bulk sorting is a add-on capability. We have a different cartridge for it. It basically, the idea is that you can run, instead of 1 to 2 cells per second, you can run, say, 50 cells per second, and sort a population at a faster rate. Much like a traditional flow cytometer or FACS instrument would, although, we are much slower than a traditional, a FACS ARIA, for example, which can sort 2,000 cells per second. We're in the 30 to 50 events per second, but we have a much better single cell dispensing capability. So it's what do you need the instrument to do? So we do have that bulk sorting capability. And it basically just enriches the cell and the cells that you're trying to sort into an Eppendorf tube. And then the rare cell enrichment, you can either use the bulk sorting or the single cell cartridge with the rare cell enrichment. But the idea is that we can load a sample with a higher concentration into the cartridge, and if we turn off one of the lasers, we can process more cells through the instrument. And this would be if you want to search for, say, 1 out of a million cells. You're looking for a very rare cell. And basically, by turning off a laser, it enables the instrument to kind of run that higher density through the instrument. And as long as your cell is labeled with fluorescence, we can sort it and detect it, and put it into a tube for you. So those are the 3, I call them kind of workflows or modes, but yeah, it's a single cell dispensing, bulk sorting, and rare cell enrichment.
Right. Thank you very much. Larissa, eu não sei se a gente tem mais perguntas aí que foram enviadas durante a apresentação.
Foram essas as perguntinhas que a gente recebeu.
Tá.
Não enviaram nenhuma aqui no chat. Se alguém tem alguma dúvida, gostaria de enviar aí no chat. Bom, vamos seguir então, Laure. Gostaria de agradecer novamente, né, a todos que participaram, também a Laure e ao Ryan, thank you. Com isso, chegamos ao fim desse webinar, tá bom? Caso tenha ficado com alguma dúvida, entre em contato com a gente consegue responder todas as perguntas para vocês também por e-mail. Vou deixar aí na tela o contato das meninas, então tanto a Laure como a Gisele, que é a nossa especialista de produtos aqui na Enterprise, tem o contato delas aí na tela. Entre em contato com a gente, envie suas dúvidas, perguntas, sugestões. Se vocês não acompanharam também, os nossos últimos webinars estão todos disponíveis na nossa plataforma através do nosso site. Então, escaneando o QR Code que está na tela, você consegue acessar todos os webinars que já fizemos dessa série, tá bom? Obrigado novamente, pessoal. Tenham um ótimo dia. Acompanhe nossas redes sociais e fique por dentro dos próximos conteúdos, que tem bastante evento vindo por aí, tá bom? Esperamos nos próximos eventos. Até a próxima. Obrigada novamente, Laure.
Thank you. Thank you, Ryan. Thank you, everybody. See you the next time.
Sounds good.